Hi, this is Mr. Anderson, and welcome to the Chapter 13 podcast on how populations evolve. This one, I'm really going to talk about the Hardy-Weinberg equation and get into the nuts and bolts of how populations actually evolve. Remember this, the smallest unit that can ever evolve is a population. In other words, this blue-footed booby is stuck the way he's made. But this population has the ability to change, but that's built on the backs of surviving or dying blue-footed boobies. What do I mean by that? This is a famous study that was done on the ground finches of the Galapagos Island. This is by Peter and Rosemary Grant. And what they studied was the beak depth of ground finches, and they found that during dry years the average would actually go up. In other words, their beaks would get a little broader, and then during wet years they would get smaller. This kind of makes sense if you think about it. During a dry year it's hard to crack open seeds, and so you want a beak that's a little bit bigger. It's kind of like a bigger pliers. You can open them up. This one is maybe not as intuitive. Why would you want a smaller beak? Well, think about that. It's hard to make a big beak. It's heavier. You got to fly it around. So there are disadvantages to having a big beak as well. And so there are pressures on either side. And so that kind of makes sense. These individuals are getting bigger and smaller bird beaks. Until you really look at the data, you don't have an understanding of how populations can evolve. And so this is the actual numbers. You can see this beautiful bell-shaped curve of the data. Here's the average right down the middle. In 1976, they found 751 birds, all the birds they could find on that island. In fact, all the birds on that island. And so it represents this nice bell-shaped curve. Here we are in 1978, post-drought. We see the shift in this towards the big, in other words, towards a bigger beak depth. But it's more telling if you actually look at the N numbers. So if we look at the N numbers, the total number of birds they found in 1976 is 751. In 1978, they only found 90 survivors. And that means that most of them actually died. Hundreds of these medium ground finches died. Which ones died? The ones that happened to have a smaller beak depth. And as they died, the population changed and moved in that direction. So we call that directional selection. And it's one of three ways that populations can evolve. What happened was the beaks of those smaller finches, the finches with smaller beaks died, and the whole population moved in this direction. We can also have stabilizing selection. That actually made those beak depths the way they were. Example in humans might be average birth weight in humans. If you're too small, let's say you weigh 2 pounds when you're born as a human, you probably didn't survive. And if you were 14 pounds, you probably didn't even get out. And so we get this stabilizing selection of babies that are around 7 pounds when you're born. We can also have pressure in the middle, and we'll talk more about this in the next chapter, that can actually divide a population in two and can create two new populations. So before we get into that, we should talk about a gene pool and what a gene pool is. You kind of know, but in biology what a gene pool means is it's the sum total of all the alleles in a population. And so alleles, remember, are the different variations of a gene. The way I like to think about that is it's like a deck of cards. So a gene pool is a deck of cards, and each of those cards represents one allele. And so let's say in here that we have two alleles. We've got the red allele, and then we've got the black allele. And let's say this stands for whatever. So like sickle cell anemia, for example. And so what sex does is sex essentially shuffles up that deck of cards and then mixes it up for the next population. So I deal this out in groups of 2 to each individual. Those individuals then interbreed as well, and then over time the gene pool doesn't really change. The genes just get passed from generation to generation to generation. Now individuals die, but the cards get passed on over and over and over and over. So let me give you like a real world example of that. Let's say I've got a deck of cards here, which I do, and there are 20 cards inside it. In here we'll say that the black cards represent the dominant allele. This could be, for example, I don't know, like a hitchhiker's thumb that bends backwards, and then the red is going to be the recessive allele. And so I've got 20 total cards here, so 20 alleles. This would be 10 individuals. And let's say that I've got, which I do, 16 black cards and 4 red cards. Well, you can do the math in your head, but 16 divided by 20 is going to be equal to 0.8. And so Hardy and Weinberg are these two mathematicians who come up with this great equation that in my class you have to memorize this, both of these equations here. But what they discovered is that p-value is going to be equal to the dominant allele frequency. And so remember, 16 divided by 20 is 0.8. And then to do the allele frequency of the recessive, 4 divided by 20 is going to be 0.2. And so our dominant allele, P, is going to be 0.8. Our recessive allele, Q, is going to equal to 0.2. So if we multiply those, and so let's do that. Let's say that we have some mating, and those alleles get mixed up in the gene pool, and so now we've got all these genes mixed up in here. We can actually predict with pretty good certainty what are the odds that I'm going to pull out one of these 3 different combinations. So I could pull out a black and a black, I could pull out a black and a red, or I could pull out a red and a red. What are the odds of me pulling out a black and a black? That's going to be this term right here. So if the allele frequency of the dominant is 0.8, the odds of me pulling out a black and a black is going to be 0.8 times 0.8, or 0.64. So there's a 64% chance that I'm going to pull out a black and a black when I draw 2 random ones. 2PQ is going to be 2 times 0.8 times 0.2, or that's going to be 0.32. So there's a 32% chance that I'm going to pull out one that's black and one that's red. And then what are the odds that I'm going to pull out one that's red and one that's red? Well, it's a 0.04, 0.2 squared. So it's not very often that I'm going to do that. So if I pull out the first 2 cards, the first 2 cards are black and black. What are the odds of that? It's actually 0.64. Next 2, black and black. That makes sense. In other words, 2/3 of the time they should be black and black. Next one, here's a black and a red. What are the odds of that? About 1 in 3. What are the odds of me ever pulling out a red and a red? Would only be a 4 in 100. And so the odds of that are going to be really, really low. There's a black and a black. There's a black and a red. There's a black and a red. There's a black and a black, and there's a black and a red. And so that, I don't know what the numbers worked out to, but in a population, as we deal those out each generation and then remix them to the next generation, Hardy-Weinberg predicts that it should stay about the same. In fact, there are only 5 things that can actually change that. And so let's say I have this big gene pool right here. And again, the cards represent alleles in that population. What could possibly change it? Well, one big thing could change is if instead of dealing out all the cards, let's say I only deal out 4 cards. Let's say that only 2 individuals survive. Now I've got, man, all red. So what happens? That next population is going to be built on this, and we call that a bottleneck effect when we choose a smaller amount. And so in order to maintain Hardy-Weinberg, you have to have a large population size. You also can't have gene flow. In other words, if I were to have this deck of cards and then grab a new card from another population and put it in here, that would change my frequencies as well. And so Hardy-Weinberg only stays the same if we maintain these 5 things. And you can't have gene flow in. You can't have immigration or emigration. Another thing that could change it is if I were to just change the card. If I were to change this one from a black club to a red queen, if I were to do that, that's going to change that the allele frequencies as well. And so example of that, this is a curly-eared cat. It just showed up in one cat. It's a mutation, and that's going to clearly change the allele frequencies. We also have to have random mating. In other words, I can't just like deal these out. Oh, here's a black and a black, so I'll deal that here, or here's a red and a red. In other words, you have to have mating that is totally random. And if you don't have that random mating, then you're not going to maintain that Hardy-Weinberg equilibrium. So why are we talking about math? Why are we talking about genetics when I thought we were supposed to be talking about evolution of populations? And we are. The 5th thing that can cause change in allele frequencies is going to be natural selection. And so scientists use Hardy-Weinberg to look at a population, and if they see the allele frequencies changing, but all the other things are held true, they've got a large population size, no immigration, no emigration, no mutation, make sure that we have random mating, then we know that natural selection is work, and natural selection is changing that population. And the interesting thing about natural selection is it's the only one of these 5 things that change or evolve populations that actually leads to adaptation. In other words, leads to organisms that are better adapted to their local environment. Thanks.